again. This video is primarily focused on the things that have happened since Darwin's Origin of Species, at least some of the major findings and events. Most scientists accepted the idea of evolution, but many were against the mechanism of natural selection that Darwin suggested. And the major problem was there was a lack of a theory of inheritance, but at that time, unbeknownst to most, Gregor Mendel was already working on that, and in 1866 published about the laws of inheritance that he'd found in Garden P. Before I talk specifically about Mendel, I just wanted to go through a little bit of historical context to put this in some sort of linear fashion for you. If we start over here with the present, then back in 2003, the human genome was determined. Sequencing of a virus back here, the genetic code. Watson and Crick discover the structure of DNA. Here's World War II back here, World War I there. Mendelian genetics is established, although Mendel's work was way back here, and that was about the same time as the Civil War was going on here in this country, and the Origin of Species was published. And then Kelvin here estimates the age of the Earth. There's Darwin's Voyage, and Lyell describes prehistoric ages in rocks. Here's Lamarck back here, so it's essentially the introduction of evolutionary theory. Declaration of Independence. There's Mozart and Linnaeus classifications, Beethoven and Bach, back to Van Leeuwenhoek's microscope. And then Ray classifies all of the plants that were known at that time and describes fossils as long dead organisms. And then establishment of the first permanent settlement in the United States, Jamestown. Proceeding further back on a different time scale, then there's the printing press, the Roman Empire, and then 2,500 years ago, this is Xenophanes and Aristotle described fossils for the first time. And then going back to Stonehenge, Mesopotamia, and then the domestication of certain animals and plants back here, and the beginning of civilization really is about 11,000 years ago. So let's move ahead again to the time of Darwin and Mendel. So here's Gregor Mendel, who was working on his Garden P experiments at about the same time that the Origin of Species was being written and eventually published. He was primarily a friar, although he wanted to be a teacher, but every time he tried to pass the exam for public speaking, he didn't pass. So he continued to be a friar, grew plants for the monastery where he lived and worked, mainly for food. And he's really pretty much the original geneticist. Published his paper on genetics in 1866, and it wasn't called anything about genetics. It was the inheritance, essentially, of characters among plants. His work was published completely in German, and not as many people spoke German in that time, especially scientists. So it wasn't read by very many people, and during the next 34 years, it was cited only 3 times, and it was only rediscovered after his death many, many years later. So he did some experiments on Garden P to try to improve the yield and nutrition, essentially, of the pea plants. Pea has the advantage of being self-pollinating, but you can also cross-pollinate it. So you can cover up the flowers once they come out and allow them to self-pollinate, or you can take the pollen from one plant, put it onto another plant or another flower, and fertilize it that way. So you can do genetic crosses, what we now know as genetic crosses. So eventually he chose several characters that bred true. That is, if you allowed the peas to self-pollinate over many, many generations, the characters bred true. And then he could cross two pea plants that had different characters and then look in the first generation and the second generation of self-pollinated plants, how those characters segregated among the progeny. So he discovered that some characters were dominant, that is, they showed up more often in the F2 generation after self-pollination of the hybrid. And there are some that were recessive, that is, they showed up less often. And the ratio was almost always 3-to-1, 3 dominant characters to the 1 recessive character. What he didn't know then, but we know now, is that each of those characters is caused by a gene that makes a certain protein product that causes those characters. And so during meiosis, those characters separate into individual chromosomes, and then when you do the cross, those alleles come together again. A dominant allele will always show the dominant character, and the recessive allele will only show that recessive character if 2 of those chromosomes or genes are present in the diploid at the end. So with Mendel's discovery that there are heritable characters that breed true, and Darwin's theory of evolution, that is, that you have the origin of species originating from some sort of mutation events, followed by diversification of a population and selection. But both of these ideas didn't come together. That is, Darwin knew nothing about Mendel, and Mendel knew nothing about Darwin. So at the time, they weren't brought together. But then in 1900, 4 people, Tschermak, De Vries, Correns, and Spillman, rediscovered Mendel's paper independently. It seems kind of amazing to me it was independent, but that's what it's reported. And this led to the establishment of the field of genetics. And so now we have genetics, which deals with the inheritance of characters and natural selection, leading to the origin of new species. And at about the same time, Friedrich Miescher first isolated something he called nuclein, which we now know as DNA. So he isolated the first nucleic acids at that time, around 1869. Also, there are increasing numbers of fossils being found and described and traded, etc., so that different evolutionary patterns are beginning to emerge among some of those fossil finds. There also are other fundamental discoveries being made, one by Louis Pasteur in the middle of the 19th century, that prove that germs cause disease and that there was no such thing as spontaneous generation. So organisms had to give rise to like organisms of the same species, and something would be contaminated or infected only if infected by certain germ species. Then in 1902, Walter Sutton and Theodore Boveri and others noted that characters followed along with how the chromosomes were inherited. So they came up with the chromosome theory of inheritance. However, at that time, it wasn't known whether the chromosome, or the part of the chromosome that was the heritable part, the part that conveyed inheritance, consisted of the nucleic acids or the proteins that were attached to the nucleic acids. Most believed that the nucleic acids, the DNA, was too simple to be the conveyor of the inheritance, and most thought that it was the proteins attached to the DNA that were the modes of inheritance. In the 19-teens and 20s, Charles Wolcott was initially walking around with his wife down a trail and found some shale that had broken off from rocks above. And he saw fossils embedded in them. And they were fossils that were very unusual. He had never seen anything like these. And so they began digging at that time, and other people started digging the fossils out and found an amazing array of animal types. More animal families existed at that time than exist today. And it was termed the Cambrian explosion, where a large array of diverse animal types appear in the Cambrian strata or Cambrian rocks at that time. It turns out that we know now it wasn't quite an explosion, but it was a fairly rapid evolution of animal types during that period of time. And that period of time was about 500 to 600 million years ago. Viruses were discovered around 1900 and after. And the first one to be discovered was tobacco mosaic virus, and all it was known was that they were smaller than bacteria. It wasn't known exactly what they were. And that wasn't discovered until much later in the 1930s and 1940s with the advent of the electron microscope, where you could actually see them, these little virus particles. Actually, at the time, influenza A was discovered as being a virus was in the 1930s. And remember, the pandemic was in the 1917 through 1919. So during the 1918 pandemic, influenza was thought to be a bacterium and not a virus. But then 10 years, 12 years later, they discovered, no, it was actually a virus. In the 1920s and 30s, genetic recombination was discovered, and one of the main people who discovered major aspects of genetic recombination was Barbara McClintock. She studied recombination in maize, so she did genetic crosses in maize, and then looked at the outcome both cytologically and morphologically. She also, in the 40s and 50s, discovered these elements that move around within the genome called transposons. And at first she was sort of ridiculed for her proposal that these were moving around in the genome. But eventually people discovered that she was correct. And after molecular biology methods confirmed this, eventually in 1983 she got the Nobel Prize. Surprisingly, in her early work, because she was female, she was not allowed to be the head of a lab, so she worked in other people's labs for much of her career. Also in the 1920s and 30s, horizontal gene transfers were first described. And since then, they've been found to be much more important during evolution than was initially thought. But in this case, it was found that certain bacteria could pick up DNA from the environment and incorporate it into their chromosomes. Also, viruses could transfer pieces of DNA, which would become integrated into the chromosome of the bacterium. And in some cases, plasmids could be transferred from one cell into another and then reside in the recipient cell. So there were quite a few discoveries made in the 20s and 30s with the advent of new methods for studying bacteria and genetics. By the 1930s and 1940s, genetics had been well established. So bacterial genetics, insect genetics with Drosophila, human genetics, and then plant genetics. So a lot of different organisms were being studied, and the evolutionary modern synthesis of evolutionary thought included genetics and selection, and specifically in the genetic part it was mutation, and then inheritance and natural selection cause adaptive evolution. And these different theories were put forth by Ronald Fisher, Haldane, and Wright. And still there are many aspects of this that are still used today in modern genetic and evolutionary projects and research. And from the 1930s to the 1960s, Dobzhansky and Ernst Mayr were very active in looking at and investigating various aspects of evolution. And they proposed that macroevolution, that is, the evolution of species up to families, etc., was caused by the same events as those that occurred in microevolution, that is, individuals, mutation leading to populations of mutant organisms up to the point of speciation. Up until the 1940s, although it was known that the chromosomes carried the hereditary material, it was not known whether it was the DNA or the proteins attached to the DNA that were the hereditary part of the chromosome. However, in a couple of different experiments, first by Avery, MacLeod, and McCarty, studying a pathogenic bacterium in mice, they found that they could transfer DNA from one strain to another to make it pathogenic. And it was only the DNA that had this effect. So it was established that the DNA was the hereditary material. In another experiment by Hershey and Chase, 1952, they used bacteriophage. And in that system, they could selectively label the proteins with radioactive sulfur and the DNA with radioactive phosphorus, and determine when they got a transformation of the bacterium by the viral DNA or by the viral inheriting factor, which one it was. And it was found definitively that it was the DNA and not the protein that was the conveyor of the hereditary traits. In 1952, Joshua Lederberg discovered plasmids. So these were extrachromosomal, autonomously replicating fragments of DNA that were found in some bacteria in addition to the chromosomal DNA. And so now we have other parts of the cell which contain other hereditary units that aren't attached to the chromosome. So their way of changing the cell can differ depending on how many of those plasmids are around. And some of the plasmids, additionally, could integrate into the chromosome. So there were a couple different factors that were by no means Mendelian in their inheritance patterns. After it was clear that the DNA carried the inheritance factors, that is, the genes, and not the proteins, it was important to discover the structure of the DNA. And this was done, of course, by Watson and Crick, but with a lot of help from Rosalind Franklin, Maurice Wilkins, and Ray Gosling. And they reported their results in 3 back-to-back papers in 1953. So the Watson and Crick paper that everyone knows about was accompanied by 2 other papers that had all of the data necessary to make the conclusions that Watson and Crick made. And so although they got the Nobel Prize along with Wilkins, there were several other very important people involved in the discovery of the structure of DNA. And it was a double helix, anti-parallel strands of DNA running in, that is, in opposite directions, carrying the 4 bases, A, G, C, and T in the center, and the sugar phosphate backbones on the outside of the molecule. After the structure of DNA was determined, it became important to understand how the DNA was copied so that you could maintain high fidelity of the genes generation after generation. So Matt Michelson and Frank Stahl then did some experiments where they grew bacteria in heavy nitrogen and in light nitrogen, N15 and N14. So they would begin by growing the bacteria in heavy nitrogen and then extract the DNA and run it on a cesium chloride gradient. The heavy molecules, which now had two heavy strands, would go further down in the density gradient because they were more dense. And then they would grow the next generation in the light nitrogen, and they found that the band moved up, indicating that DNA now had one heavy strand and one light strand of DNA. They continued to grow the bacteria in the light nitrogen and got increasing numbers of molecules that indicated that they contained both light strands, and then there were some still with the light and the heavy strands. And eventually, after many generations, doubling times of 20 minutes each, most of the DNA had two light strands, and very few of the molecules had one light strand and one heavy strand, and no molecules at the end had both heavy strands, indicating that the DNA underwent semi-conservative replication. That is, one strand was always used as a template to build a new strand that was complementary to it. Incidentally, Frank Stahl was one of my professors when I was an undergrad. He was not only an excellent researcher, but he was an excellent teacher as well. So one of the next steps in the field that became molecular biology was, how do you determine the genetic code? And this was done during the 1960s by Nurnberg, Hawley, and Corana. And they employed several different methods to determine what the genetic code was. By this time, they had discovered that the DNA was transcribed into RNA, mRNA, and that the tRNAs then attached to the mRNA in a code-like structure to then deliver each of the amino acids to the growing polypeptide chain in the ribosome. But they did it in several different ways. First was by starting with codons that were things like UUU and CCC, and then they could determine those ones. Then they went on to more difficult codons and eventually finished that off as far as the coding of the genetic coding for protein synthesis was finally completed in the late 1960s. So all these pieces are fitting together, starting with Mendel and Darwin and going into genetics during the 20s and 30s, and ultimately determining the structure of DNA and then the genetic code to understand more about how genes change, how translation works, and how that affects the evolution of the organisms. And then every once in a while, someone comes up with a strange idea that people argue against, including transposons with Barbara McClintock. But starting in the beginning of the 20th century, there were people that started to propose that organisms essentially swallowed up other organisms, and then those swallowed up organisms became the organelles. And this was synthesized eventually by Lynn Margulis, originally Lynn Sagan. She was married to Carl Sagan initially, by which an organism, a bacterium, is taken up by a protist cell with a nucleus, and then that bacteria that was taken up became a mitochondrion. And to become a chloroplast, then you have a mitochondrial protist cell with a nucleus and a mitochondrion, and then it takes up a cyanobacterial cell to become the chloroplast. Now, we know that's true because that's been studied extensively. And then more recently, others have shown that other endosymbiotic events occurred to create many different types of photosynthetic protist cells and other types, including dinoflagellates, oomycetes, water moles, and many brown algae, and many different types of organisms. And some of them are parasites, such as trypanosomes, have an organelle that came from an algal cell. I will talk about these later in the semester. It makes evolution a little more confusing, but it also makes it a lot more interesting as well. So the way it makes it more interesting and confusing is that you have a phylogenetic tree, and the root down here is already convoluted because organisms have been exchanging pieces of DNA. And then as you move your way up into bacteria, archaea, and eukaryotes, you see there are transfers of organisms that became organelles from different groups. So the mitochondria came out of the proteobacterial group to become a mitochondrion in plants and animals, and protists as well. And then the plastids entered via a cyanobacterial ancestor that made these protist cells photosynthetic. And there are many other exchanges that occur. There's just a few indicated here, but there are literally thousands and thousands of exchanges that have occurred during evolutionary time. Then in the 1960s, we have a further refinement of what happens during evolution and selection. As was synthesized in the 30s and 40s and 50s, evolution essentially consisted of mutation and selection. So Kimura decided to test that out on real organisms to see what effect selection had. And so he did many experiments to determine that most mutations had neither a positive nor a negative effect on those organisms. And so this is what's called random genetic drift, where you have mutations constantly occurring. There's no selection on those mutations, either positive or negative, and so they can build up over time. And he determined that not only was this happening, but this was the major thing happening during evolution, that most mutations, 60, 70, 80%, had no effect whatsoever on the organisms or the evolution of those organisms. Beginning in the 1970s, then we have the first recombinant DNA, that is genetic engineering, where you could take a plasmid or a bacteriophage, insert a piece of DNA that you would like to amplify or study, and grow that in a bacterium and obtain literally millions and sometimes billions of copies of those sequences for study. So this allowed manipulation of specific genes and gene regions and chromosome regions for study in evolutionary studies, in functional studies for what the gene does, and for making the proteins from those genes to determine what the proteins did in the cell as well. In Darwin's time, it was thought that evolution moved in these slow, continuous, gradual changes. And so on the bottom, this tree shows that, where you have gradual changes leading to extinction and speciation up to the present day. What Gould and Eldridge found and proposed was that evolution goes in fits and starts. That is, you have these sudden bursts of evolution, sudden bursts of change, followed by stasis, where the organisms change very little. This had been proposed back in the early 1900s also, but there was no distinct proof that this was actually happening. But Gould and Eldridge proved in a number of studies that this was at least one of the ways that evolution was proceeding. By the mid and late 70s, sequencing methods were finally developed, first on RNA, and this sequence was actually quite short for virus MS2, but it took several years to accomplish. So this was in 1976. The first RNA genome was sequenced. By that time, DNA sequencing methods had not yet been invented. The next year, 1977, Fred Sanger and Walter Gilbert invented two different ways to do DNA sequencing. The Sanger method became the more popular and easier to accomplish. The Gilbert method was a degradative process, while the Sanger method was a polymerization reaction, so it worked much better, and you needed less DNA to sequence. But these were the first sequences that were accomplished ever. Some of the first sequences indicated that the genes were discontinuous in some cases. This is also confirmed by electron microscope studies, as well as some other informational studies. And so you have exons, which became the expressed portions of the gene, the RNA. And then there were introns, intermediate intervening sequences in between the exons that were spliced out before the mature mRNA was produced. And furthermore, you could have alternative splicing, which was common in viruses and also in certain animal genes and sometimes plant genes, where a single RNA could be spliced in different ways to produce different mRNAs and thus produce different proteins. At about the same time, people were studying fossils from different layers in the rock. And they had discovered that about 66 million years ago, there was a mass extinction. And one of the casualties of that mass extinction was all of the dinosaurs, which had existed for about 200 million years, and suddenly they were gone. So the Luis and Walter Alvarez, a father-son team, started looking at what was unique about that rock layer. And they discovered by looking at how much iridium was in the rocks that above that 66 million mark, there was very low iridium. And below that, there was very little iridium. But right in that layer, there was a high concentration of iridium. And iridium is rare on the Earth, but it's fairly common, or at least at higher concentrations, on meteorites and asteroids. So they proposed that this mass extinction was caused by an asteroid strike on the Earth, and a pretty substantial asteroid strike. And years later, it was discovered that on the Yucatan Peninsula near the town of Chicxulub, there was a crater, and the age of that crater, as it was dated, matched at about 66 million years ago that it struck the Earth. And so it's pretty firm evidence that that caused the extinction of the dinosaurs and many other organisms as well. Other people were studying the mass extinction at 66 million years ago, as well as others, including Sapkowski and Raup, and they, from fossil data, determined that there were 5 mass extinctions, including the one 66 million years ago, and that one wasn't even the most major one. The most major extinction, which wiped out about 90% of all species, happened about 252 million years ago. But there have been mass extinctions in the past, and whether they're all caused by asteroids or caused by other factors is still somewhat uncertain. Asteroids, volcanism, gamma ray bursts, things like that have been proposed for these different mass extinctions. Although sequencing had been available since about 1977 to labs. Mainly people were working on individual regions of the chromosomes or single genes. But the first DNA genome that was fully sequenced was that of FIX174. And it was also examined as far as using mutants and how it was put together and what happened during infection. So a lot of study went into this FIX174, which helped a lot to determine the type of expression from these genes that occurred to make this virus infectious and make new particles and how it functioned within the cell. So that was in 1984. Up until the 1980s, people were amplifying their DNAs within plasmids in bacteria. So that's the way they would get more copies of the DNA for sequencing and for functional assays, etc. But in early 1980s up until 1985, Kary Mullis developed what's called the polymerase chain reaction. And that was based on some experiments by Gobind Khorana back in the 1960s and 70s. And using a target DNA, you could then produce primers for each end and use a DNA polymerase to heat and cool and polymerize copies of that target sequence over and over and over again. So you didn't need to use bacteria to make more copies of the fragment that you were interested in. You would just do it in a test tube, in a small test tube, in a PCR reaction. By the way, Kary Mullis describes how he came up with the idea in, I think it was a Scientific American article, where he was out in the California desert driving around at night after having taken some mood-altering substance and just saw these molecules amplifying in front of his eyes at the time. And so luckily he made it back in one piece from the desert, came up with the idea, and the PCR reaction has been used billions and billions of times around the world now. Around the same time, Altman and Czech discovered that there were certain RNA molecules that had enzymatic activity. Now, up until that time, all enzymes were thought to be proteins, but they discovered that there were certain ribozymes, is what they called them, and some of these were self-splicing introns, group 1 and group 2 introns in mRNAs, would splice themselves out. There was also something called the hammerhead ribozyme and others. This led to some hypotheses about the first organisms on Earth, and now it's thought that the original organisms, the very first organisms on Earth, were based on RNA and probably some peptides, rather than DNA and proteins. In 1995, the first complete bacterial genome, that of Haemophilus influenzae, was sequenced. So that was quite an undertaking at the time. It was over a megabase in size, so it's quite a large number of nucleotides to sequence, and it took several labs, several years to complete the project. Today, right now, you can do an entire bacterial genome in a few hours. That's how they're done today. But at that time, this was quite an undertaking to get this first one done, and then to characterize the genes that they found in that genome. A year later, the first eukaryotic genome, that of Saccharomyces cerevisiae, was determined. This was quite an undertaking. It's about 6 to 8 times larger than the Haemophilus influenzae genome at about 12 million base pairs of DNA. So it was quite an undertaking. There are 16 chromosomes. Each one was sequenced by different labs and then put together in the final genome and published in 1996. In the early 2000s, microbiomes were starting to be characterized. And so this picture just shows the different types of bacteria that are living on a person, both male and female, and the various types of organisms that live in those different areas on your body. So you can think of any animal, or any plant for that matter, being an ecosystem for different types of organisms, and they will inhabit different parts of the body. And if you have some types of microbiomes, they will protect you against pathogenic organisms. So these are not just happen to be living there. They've been living with humans and other animals for a very, very long time, millions and millions of years, and have evolved to grow in those different areas. And the humans, the larger organisms, have evolved to be sure that they have those organisms on them. So there's been this co-evolution of the microbes and the macroorganisms, such as humans, dogs, cats, you name it, chickens, anything, as well as plants and other organisms, all have their specific microbiomes living on and in them. In 2003, the first draft of the human genome was completed by two different groups, Craig Venter's group at Celera and Francis Collins' group at the National Institutes of Health. So one was a private company and one was a public university research center. And they were done in two different ways. Collins sequenced just the expressed versions of the genes, that is, the genes themselves, which constitute only about 1.5% of the total genome, whereas Venter did it a different way. He chopped up the genome in many different pieces and then sequenced all the separate pieces randomly and then threw them into a computer. And the computer looked for overlaps and stitched together each of the chromosomes. So he got more of a complete version of the human genome with the genes as well as the intergenic regions as well. So the entire genome, of course, consists of 22 somatic chromosomes and then the sex chromosomes for a total of 23 per haploid, or 46 for a diploid cell. And there are the number of genes on each one, the number of proteins, and you can see that the number of genes is less than the number of proteins, and that's due to alternative splicing. So each gene can produce sometimes more than one protein. And then you also can see the mitochondrial genome there, quite small, and the links of each one of the chromosomes. Shortly after that, or actually just before that, comparative genomics was started, and that's the process whereby you compare the genes in one organism's genome with those in another organism to try and find out what's the commonality, what are the common genes that are found in everything, and then what are the unique genes for specific purposes in each one. A lot has been done with comparative genomics since that period of time, and you can see here in this Venn diagram the percentage of the genes that are in common with worm, a nematode, humans, and Drosophila, a fly. About 43% of the genes are in common, and if you look at all of the genes that are in common to both those animals and fungi, amounts to nearly 70%, so that a lot of genes are in common, and then you have some specific genes out for each one of those organisms that leads to the more specific changes that result in those different body plans and life cycles and life histories. So more recently, there has been improvements in sequencing, so now there's next generation sequencing, a lot being done with genomics, metagenomics, transcriptomics, and others. to the point where sequencing is no longer a problem. You can do a lot of sequences. In fact, right now there are more sequences on GeneBank and other databases that have never been completely analyzed, and so you can now just do a lot of data mining. You don't even have to do much lab work. You can go straight to the databases and do all sorts of projects now. Okay, so that's it for this video. We'll start a different video for next time. Thanks.